Good morning, everyone. I am Dr. Kalpana Sharma, Associate Professor, Department of Zoology, S.P.C. Government College, अजमेर। बच्चों, मेरे लास्ट के लेक्चर में मैंने आपको न्यूक्लिक एसिड्स के बारे में बताया और स्पेशली आरएनए के बारे में मैंने डिस्कस किया। आज के लेक्चर में मैं आप लोगों को डीएनए के बारे में बताने जाऊंगी। What is DNA? Deoxyribose Nucleic acid. Now, first of all, the question arises, what are nucleic acids? Nucleic acids, naturally occurring chemical compounds that is capable of being broken down to yield phosphoric acid, sugars and a mixture of organic bases. Nucleic acid क्या होते हैं? Naturally occurring carbonic compounds से, organic compounds से, जो जिनका breakdown किस में हो जाता है? Phosphoric group, sugars and a mixture of organic bases, purines and pyrimidines. Nucleic acids are the main information carrying molecule of the cell. And by directing the process of protein synthesis, they determine the inherited characters of every living things. The two main classes of nucleic acids are DNA and RNA. DNA is the master blueprint for life and constitutes the genetic material in all free living organisms and most viruses, whereas RNA is the genetic material of certain viruses. But it is also found in all living cells, where it plays an important role in certain processes such as the making of proteins. DNA stands for Deoxyribose Nucleic acid, which is a molecule that contains the instruction an organism needs to develop, live, and reproduce. These instructions are found inside every cell and are passed down from parent to the children. It is a nucleic acid and is one of the four major types of macromolecules that are known to be essential for all forms of life. DNA is found in nucleus, with a small amount of DNA also found in mitochondria in eukaryotes. In this picture, you can see the structure of a deoxyribose sugar, purines, adenine and guanines, and pyrimidine, cytosine and thiamine, and also the structure of the uracil, which is present in RNA, as I told you earlier. In this diagram, you can see the structure, double helical structure of a DNA molecule, which was given by Watson and Crick. Now, I will discuss in detail about the structure of DNA. Watson and Crick, they proposed the double helical model for DNA. The sugar phosphate backbones are the outside of the double helix, and purines and pyrimidines from the rugs of the DNA helix ladder. DNA में क्या होता है? बाहर की तरफ बैकबोन्स तो है इसकी शुगर और फॉस्फेट, और रग्स जो सेंटर में क्या है इसके? प्यूरिन्स एंड pyrimidines. The two DNA strands are anti-parallel to each other. ये जो दो strands हैं वो एक दूसरे के anti-parallel होते हैं. The third one is the direction of each strand is identified by numbering the carbon in each sugar molecule. कैसे numbering होती है? The 5' end is the one where carbon #5 is not bound to another nucleotide, and the 3' end is the one where carbon #3 is not bound to another nucleotide. In 1953, James Watson and Francis Crick, they discovered the structure of DNA. The work of Rosalind Franklin led to Watson and Crick's discovery. Franklin first had pointed out that the DNA is made up of two spirals. The structure of DNA is double helix structure because it looks like a twisted ladder. डबल हेलिकल होता है जैसे एक सिड्डी दूसरी सिड्डी के ऊपर twisted रहती है. The sides of the ladder are made up of alternating sugars. In DNA, it is the deoxyribose sugar and phosphate molecules, while the steps of the ladder are made up of a pair of nitrogenous bases. There are 4 types of nitrogenous bases: adenine A, thiamine T, guanine G, cytosine C, DNA pairing. The nitrogenous bases have a specific pairing pattern. ये जो nitrogenous bases है, इनका pattern जो होता है वो specific होता है. क्योंकि हम देखते हैं कि एडेनिन इज द अमाउंट ऑफ एडेनिन इज इक्वल टू अमाउंट ऑफ थायमीन, हेंस द एडेनिन विल पेयर विद थायमीन एंड द अमाउंट ऑफ गुआनिन इज इक्वल टू द अमाउंट ऑफ साइटोसिन. द पेयर्स आर हेल्ड टुगेदर बाय हाइड्रोजन बॉन्ड्स. In this picture, you can see the diagram of a ribose sugar. The sugars, as I told you, are the backbones of the DNA molecule. Deoxyribose is a modified form of another sugar called ribose. I am going to give you the structure of that first because you will need it later anyway. Ribose is the sugar in the backbone of RNA, ribonucleic acid. This diagram is showing that deoxyribose has a hydrogen here rather than OH group. This diagram misses out the carbon atoms in the ring for clarity. Each of the 4 corners where there isn't an atom show has a carbon atom. The heavier lines are coming out of the screen or paper towards you. In another word, you are looking at a molecule from a bit above the plane of the ring. So that's ribose. Deoxyribose, as the name might suggest, is ribose which has lost an oxygen atom, deoxy. This is a diagram. The only other thing you need to know about deoxyribose or ribose for this matter is how the carbon atom in the ring are numbered. The carbon atom in the ring are numbered in which pattern? The atom to the right of the oxygen, as we have drawn the ring, is given the number 1. And then you work around to the carbon on the CH2OH side group, which is numbered 5. हम OS group से number 1 शुरू करते हैं और जहां CH2OH group है वहां पे numbering 5 end होती है. You will notice that each of the number has a small dash by it 3 dash or 5 dash, for example. If you just had ribose or deoxyribose on its own, that wouldn't be necessary. But in DNA and RNA, these sugars are attached to another ring compounds. The compounds, the carbons in the sugars are given the little dashes so that they can be distinguished from any numbers given to atoms in the other ring. You read 3 dash or 5 dash as 3 dash prime or 5 dash prime. Now the second one is attaching a phosphate group. फॉस्फेट ग्रुप कहाँ पे अटैच्ड होता है? ये शुगर के आप देख रहे हैं इसमें फॉस्फेट ग्रुप का अटैचमेंट कैसे होता है? द अदर रिपीटिंग पार्ट ऑफ द डीएनए बैकबोन इज द फॉस्फेट ग्रुप. अ फॉस्फेट ग्रुप इज अटैच्ड टू द शुगर मॉलिक्यूल in place of hydroxyl group on the 5' carbon. ये 5' कार्बन एटम का जो OH ग्रुप होता है वो रिप्लेस हो जाता है फॉस्फेट ग्रुप के लिए. Attaching a base and making a nucleotide. देखिए न्यूक्लियोटाइड, जैसे मैंने लास्ट लेक्चर में भी बताया था कि शुगर प्लस फॉस्फेट प्लस नाइट्रोजनस बेसिस। तो इस पिक्चर के अंदर आप क्या देख रहे हैं, इसके अंदर शुगर भी है, इसके अंदर प्यूरिन्स पायरिमिडीन्स भी हैं और इसके अंदर फॉस्फेट ग्रुप भी है। तो ये कंप्लीट न्यूक्लियोटाइड है। द फाइनल पीस दैट वी नीड टू एड टू दिस स्ट्रक्चर बिफोर वी कैन बिल्ड अ डीएनए स्ट्रैंड is one of the four complicated organic bases in DNA. These bases are cytosine, thymine, adenine, and guanine. Note, these are called bases. Why they are called bases? Because that is exactly what they are in chemical term. They have lone pairs on nitrogens and so can act as electron pair donor. बेस क्यों कहलाते हैं ये? क्योंकि ये जो होते हैं केमिकल टर्म के अंदर ये क्या होते हैं, इलेक्ट्रॉन पेयर डोनर्स होते हैं. दे आर इजंट पर्टिकुलरली रेलेवेंट टू द फंक्शन इन डीएनए, बट दे आर ऑलवेज रेफर्ड टू एज बेस anyway. These bases attach in place of the OH group of the 1' carbon atom in the sugar ring. देखिए यहां से हमने नंबरिंग शुरू करी थी, जहां पे 1 कार्बन एटम होता है वहां पे बेस अटैच होता है और 5 कार्बन एटम वाली पोजीशन के OH ग्रुप को रिप्लेस करता है फॉस्फेट ग्रुप. Here in this diagram, you can see the structure of the purines and pyrimidines, cytosine, thiamine, adenine, and guanine. The nitrogen and hydrogen atoms shown in blue on each molecule shows where these molecules join to the deoxyribose. In each case, the hydrogen is lost together with the OH group on the 1' carbon atom of the sugar. This is a condensation reaction. Two molecules joining together with a loss of a small one, not necessary water. For example, here's what the nucleotide containing cytosine would look like. See here. Joining the nucleotide into a DNA strand. अब हमारे पास न्यूक्लियोटाइड बन गया, अब डीएनए स्ट्रैंड में कैसे जुड़ेगा? पहले हमने राइबोस शुगर का देखा कैसे होती है, फिर हमने देखा कि उसके अंदर फॉस्फेट ग्रुप का कार्बन एटम की पोजीशनिंग कैसे है, फिर देखा बेस कैसे जुड़ते हैं, है ना. अब देखेंगे ज्वाइनिंग द न्यूक्लियोटाइड इनटू अ स्ट्रैंड. स्ट्रैंड के अंदर कैसे जुड़ता है? अ डीएनए स्ट्रैंड इज सिंपली अ string of nucleotides joining together. I can show how it happens perfectly well by going back to a simpler diagram and not worrying about the structure of the base. The phosphate group on one nucleotide linked to the 3' carbon atom on the sugar of another one. In the process, a molecule of water is lost, another condensation reaction. Thank you.